Welcome back from the break. Let's take a look at the concept check question and then move on. So compare and contrast your nutritional mode with the nutritional mode of a typical fungus. So remember, as an animal, you are a heterotroph, but you are an ingestive heterotroph. That means that you ingest large particles of food and you break them down internally, beginning with chewing in the mouth, mixing with saliva and the enzymes in saliva, passing the food on into your stomach, which continues the breakdown, particularly of proteins, and then on into the small intestine, which continues the breakdown of carbohydrate, protein, and begins the breakdown of fats. And it's all done internally through enzymes that are secreted into your digestive tract, at various points along the way to break down the food. And then you absorb the digestion products into your body out of your digestive tract. Fungi, remember, are absorptive heterotrophs. They are heterotrophs just like we are, but they are absorptive heterotrophs, which means rather than ingesting large particles of food, they secrete their digestive enzymes into their environment and carry out the largest part of the breakdown of those large molecules in the environment before they are absorbed into the body of the fungus. So in this case, digestion, or the vast majority of digestion, takes place externally to the body of the fungus, and then the products of that digestive process are absorbed into the body of the fungus. So those are the differences between animal digestion and nutritional mode versus a typical fungus. So let's move on and talk a little bit more about some features of fungi. So in terms of fungal reproduction, the vast majority of fungi reproduce either sexually or asexually, and they will have within their life cycle a sexual stage and an asexual stage. And it is typically the sexual stage that defines which clade the fungi belong to. Now, this is not a uniform observation, as you'll see. There is one clade of fungi that only reproduces sexually. As far as we can tell, and this has been on the basis of much observation, there is no asexual stage. That does not still mean that we don't define them by the sexual stage, because that's all they have, asexual stage, and that allows us to define them. There are also some fungi that reproduce predominantly asexually, and we typically refer to those as molds, and we'll talk about them. They belong to one of the major clades of fungi, typically. But what we say is that while we may not know what their sexual reproductive stage is, there is likely an asexual stage. We just have not observed it yet. So many of these fungi are referred to as molds. We don't assign them specifically to a clade. There have been at times the use of a tentative phylum name for these, but as you'll see, we don't typically use it. There have been a number of different descriptive terms associated with these types of fungi. Many of them have fallen out of favor now because we still feel that even if we can't identify the sexual stage, there is one, we just haven't seen it yet, and they may not utilize it except under very exceptional circumstances. Yeasts are actually included in this because yeasts very rarely reproduce sexually. But when we get to the clade that yeasts are assigned to, I'll explain why we believe yeasts should be assigned to that particular clade, because yeasts are actually assigned to one of the major fungal clades. So here is the life cycle, the general life cycle of fungi. And you can see that there is a sexual cycle and an asexual cycle. And the asexual cycle is relatively easy to understand, because the first thing that you should recognize based on the color coding in this diagram is that the mature form of the fungus, the mycelium, made up of the hyphae, is haploid. So a fungus, when you see it in the environment, is haploid. So when you eat a mushroom, you're eating something that has a large body of haploid tissue, and we do refer to it as being functionally haploid. There's a story behind that. However, within that mycelium, if they're going to reproduce asexually, specialized hyphae give rise to a sporangium in which spores are going to be produced. They're going to be produced from haploid nuclei. The structure is haploid itself. And those nuclei are going to become encapsulated in environmentally resistant hydrophobic coatings, which allows the spores then to be released into the environment. If they land in a suitable environment, meaning moisture and a usable food resource, the spores germinate and the resulting cell begins to grow into new mycelial mass, either by creating multiple hyphae or, depending on the type of fungi, a large, let's just say a large hyphal mass that represents the mycelium. That's probably the simplest way of putting it. If they're going to reproduce sexually, then the situation becomes a little bit more complicated. Here we now reintroduce the terms that I introduced you to back when we were talking about protists, and the notion of fusion of cells without fusion of nuclei. The two terms are plasmogamy and karyogamy, and here we use them again in the life cycle of fungi. So when you have a sexual reproductive stage, it requires hyphae from two fungal masses, and we talk about opposite mating types. So here we have the plus and minus mating types again. So you need a mycelium from the plus mating type and a mycelium from the minus mating type. They produce specialized hyphae that grow towards each other in response to sexual attractants, pheromones, chemicals that are released into the environment that act as sexual attractants and cause these specialized mating hyphae to grow towards each other. At some point, the hyphae will fuse, and the two haploid nuclei that are present in each of the reproductive hyphae enter a common space. Now, what happens in this common space then determines how we talk about these hyphae. If the hyphae do not contain septae, they're not divided into cellular compartments, then we've referred to the hyphae as heterokaryotic, containing two different haploid nuclei and multiple copies of those nuclei in a multinucleated cytoplasmic compartment. If it is broken up into cellular compartments, then typically one copy of each haploid nucleus occupies every compartment. So each compartment contains two haploid nuclei, one from each of the two mating hyphae. And under those circumstances, we refer to this as a dikaryotic hyphen, meaning two different haploid nuclei. And depending on the type of fungus determines how long the haploid nuclei remain as discrete haploid nuclei. But at some point, those haploid nuclei will form by the process of karyogamy to give rise to a diploid nucleus that forms the zygote. That almost immediately undergoes meiosis. The diploid stage here is very, very transitory. And so the diploid nucleus undergoes meiosis, gives rise to haploid nuclei, which then get packaged into spores and get released into the environment where, again, if they land in a suitable environment, they will germinate and develop into a new mycelial mass. So you can see that in both the asexual and the sexual stage, spores are the reproductive structure that's released into the environment. and spores are always haploid, which means that the resulting fungus is always haploid. But in the sexual stage, there is going to be a transitory diploid stage, and it's the nature of that, of the origin of that transitory diploid stage, that really defines what type of fungus you're looking at. That's why we pay attention to the sexual side of the reproductive cycle, because that's where the differences lie, and that gives us a means of defining the clades and defining which clade a fungus belongs to. So I've talked in some detail in the diagram about all of this. So if you want to read up some more about this, it's in the textbook, or you can pull the PowerPoint. It's in the PowerPoint that's available through Canvas. And again, we talked, here I should, seeing as how I've got it here, I should talk about what we use to define those that we have yet to recognize a sexual stage. So remember, we refer to these as molds. So one of the old terminologies that's been used for an asexually reproducing fungus is fungi imperfecti, imperfect fungi. This is somewhat of a, I've used the term arrogant term previously. I feel a little bit more uncomfortable about using that way of describing this these days. But the notion of imperfection lies with the fact that animals reproduce sexually. And traditional taxonomists in the past have considered that sexual reproduction was the way to do it because it was the way we did it. And so any organism that didn't use sexual reproduction, in other words, reproduced asexually, was somehow imperfect. And so these fungi got labeled as imperfect fungi, or in terms of Latinizing that, fungi imperfecti. There was a point in time when there was a consideration of creating an actual phylum to put these in, and they became known as Deuteromycota or Deuteromycetes. We don't use that term anymore. If you see that, please understand that it is not considered to be taxonomic any longer. It has no taxonomic value. It is just a generalized label that is applied to those fungi that appear to reproduce predominantly asexually and have no recognizable sexual reproductive stage at present. Doesn't mean they don't have one. It just means that we have not recognized what that reproductive stage is. So these are the organisms that have traditionally been referred to as molds. See, yeasts are mentioned here, but as I said a little bit later in the presentation, I'll identify the clade that yeasts have been tentatively been assigned to on the basis of what they do when they do reproduce sexually, but more precisely, the way they actually reproduce asexually, which is very similar, almost identical to one of the clades of fungi. And that is the reason why yeasts are typically assigned to it. But more of that to come. So here you can see a couple of fungi that are reproducing asexually. This is actually yeast. Oops. And so you can see that the yeasts are budding off. And in fact, I might as well mention this now. What this is, you can see the bud here. This is called a conidium. And it's an asexual spore. So what you're looking at here is a structure called the conidiophore. and it is an asexual structure. And you can see on the tip little buds that look very much like the bud on the yeast cell. And that's because each of these little buds are conidia. That's the plural. So the singular is conidium. And so this particular mold growing on the surface of an orange, and I'm pretty sure most of you have seen this because it happens all too frequently, even if you leave the orange in the refrigerator, that delays the time that the fungus will develop, but it almost certainly will develop on the surface of the orange if you leave it sit around on the kitchen bench for any length of time, because the spores for this particular fungus are present in the air all around us. That's one of the things that we choose not to think about very much, but fungal spores are in the air all around us all the time. They're on our skin, we breathe them in. They're just a fact of life that they are present. And this particular fungus, this mold, does reproduce asexually via the conidiophore and the conidia. Yeasts produce a similar sort of asexual bud, and so yeasts are assigned to the same fungal clade that this particular fungus happens to be assigned to as well, on the basis of the presence of these conidia as asexual reproductive structures. So we'll take a quick break here for a concept check. I'm not going to stop the recording this time, just talk about the concept check question, because we've got a couple of short sections in here. Considering the haploid and diploid stages of the respective life cycles, how do the life cycles of a fungus, I should say fungus there, not fungi, and a typical animal differ? Well, a couple of differences. Number one, the product of meiosis in an animal life cycle are the gametes, not spores, and the diploid stage is dominant, whereas the haploid stage is really transitory. In a fungus, there is no recognition of a sperm and an egg. What you have is a pair of haploid nuclei that are derived from the mating types of fungi that are haploid themselves. So the body of a fungus is haploid. The dominant stage of a fungus life cycle is haploid. The diploid stage is incredibly transitory, and meiosis gives rise to spores as the reproductive structure that is released into the environment. So in terms of haploid and diploid, there is a different emphasis. In animals, the emphasis is on diploid with a transitory haploid stage represented by the gametes that are the product of meiosis. In fungi, the emphasis is on the haploid stage with a long-lived stable haploid stage and even a long-lived reproductive stage where individual haploid nuclei derived from the two mating fungi can remain in a stable state for great lengths of time before eventually they merge to form the very short-lived transitory diploid stage that gives rise to spores that then germinate to give rise to the next generation of fungi. So different emphasis on the haploid and diploid. So let's continue on, again, looking more at some details about fungi. So the origin of fungi lies with a common origin with animals. We believe that both animals and fungi and the protists to which they are most closely related are descended from a single common flagellated ancestor. Because remember that from our look at protist diversity and origins of eukaryotes, the fungi and animals and their related protists are assigned to a clade called opisthecons. So episto refers to the fact that the flagellum is located posteriorly. Excuse me. And the other thing that we need to remember is that in a larger sense, the epistoconts are assigned to the supergroup known as uniconta, which also makes them unique. So let me bring the arrow down here. So the two important things here are that the fungi and animals are part of the same major clade on the basis of DNA evidence called the epistoconts, where the flagellum is located at the posterior of the cell, and it's a single flagellum on the basis of being assigned to the supergroup uniconta, uni1 conta referring to flagellum. So a single flagellum located posteriorly to the flagellated stage that represents the reproductive stage or a potential reproductive stage for both animals and fungi. Now, remember at the beginning, I said the traditional definition of a fungus was that there was no flagellated stage. And that still remains largely true, because the majority of the clades that we will encounter as we look at the diversity of fungi consist of fungi without a flagellated stage. But given that the protist relatives of fungi and the basal groups that are thought to be part of fungi, and certainly chytrids, which are true fungi, all have a flagellated stage, that reinforces the fact that they are descended from a flagellated protist ancestor. And if we go back far enough, it's a flagellated protist ancestor that is shared in common with animals. But at some point during the phylogeny of fungi as a kingdom, the flagellated stage was lost. And that has been the subject of a lot of discussion. There seems to have been some resolution based on the current taxonomy as to when the flagellum was actually lost, or the flagellated stage was actually lost. Previous phylogenies hummed and hawed a little bit about exactly when the flagellum was lost. There was some suggestion that maybe the flagellum might have been lost several times during the phylogeny of the the fungal kingdom, but it appears that there is now a consensus that the loss of the flagellum consists of a single event that lies between chytrids and the rest of the fungal kingdom. More to come. So this is basically the phylogenetic tree of the epistoconts, and you can see here that animals and fungi are relatively closely related. Nuclearids, by the way, are amoeboid-like cells that feed on bacteria and things like that, but at the nucleic acid level, at the DNA level, they share a apparent common ancestry with fungi. They are the most closely related group of protist fungi, and so are considered to be not a basal group of fungi, but a closely related sister clade to fungi. So when did the divergence between animals and fungi occur? Probably somewhere between 1 and 1. I've got 1 and 1.5 million. That should be billion. Let me edit that on the fly. So it could be as far back as 1.5 billion years ago, maybe as recently as 1 billion years ago, but somewhere in that time frame, based on molecular clock evidence, is likely when the divergence occurred. We will probably never know exactly when, because the fossil evidence that's available for fungi is much younger than that. The earliest clearly definitive fossil fungi date to about 440 million years ago. Here's a photograph. You can find this in the textbook. And when you look at this and you compare this with the chytrids, these look remarkably like chytrids, or at least very similar to some modern chytrids. And that's one of the reasons why these are considered to be definitively fossil fungi. But these date from 440 million years ago. So that is a lot more recent than when we think the divergence between animals and fungi occurred from their common ancestor. So we may never truly know when fungi and animals diverged from their common ancestor. All we can say is that by 440 million years ago, we definitively have fungi that we can identify through the fossil record. When did fungi become colonizers of land? Probably at the same time as the early members of the plant kingdom, or the early ancestors of the plant kingdom, also began the colonization of land. Bear in mind that the most basal groups of the plant kingdom today, the bryophytes, lack vascular tissue and therefore don't have roots, don't have shoots, don't have leaves as we recognize them, because you have to have vascular tissue for that. They do have rhizoids, which are extensions of epidermal cells that penetrate into the substrate and allow them to anchor themselves, and to some degree can provide a way of capturing water and some nutrients. But when we look at these plants, what we do notice is they have a symbiotic relationship with fungi. And those fungi are serving the function of roots. They're aiding those plants by mobilizing more water and more nutrients, more minerals out of the soil than the plants are capable of doing, because they lack roots to do it. And because of the ability of fungal hyphae to penetrate into large volumes of the soil, and also to grow very rapidly to penetrate large volumes of soils than the roots of plants could ever penetrate, this association between fungi and plants becomes really important, and no more important than the early ancestors of the plant kingdom, seeing as how they lacked roots and lacked any mechanism for mobilizing materials out of the soil. Having a symbiotic relationship with fungi that accomplished that was obviously an advantage as they began colonizing land. And we believe that this association, as we see it in the plant kingdom today, with, as I said earlier, 90 to 95% of all plants having some sort of symbiotic relationship with fungi, represents this ancient association between fungi and plants as co-colonizers of dry land, roughly around 470 to 500 million years ago, somewhere in that time frame. So another quick break, because we have not been going for very long. Just a check on an important concept. Most fungi lack a flagellated stage. Very true. Yet fungi are classified as episthokonts, meaning a posterior flagellum. We also talked about that. Explain why. And the reason why is because at the DNA level, they are most closely related to animals, and the choanoflagellates, the protists that animals are most closely related to, they do form a clade at the DNA level. And so we do see the evidence of the flagellum in the most basal groups that are assigned to kingdom fungi, the chytrids particularly, as true fungi, and the tentative fungi, the cryptomycetes and the microsporidia, particularly the cryptomycetes, not so much the microsporidia. So at the most basal, yes, there is a flagellated stage in the basal group of fungi, and this carries back into the common ancestor of animals that lies amongst the flagellated protists. It's a single cell, a unicellular flagellated protist, and that ties them to the opisthocons. So even though the modern or most fungi lack a flagellated stage, we do see the flagellated stage in the most basal groups associated with kingdom fungi, and we also see the flagellated stage in the nuclearids, protists that the fungi are most closely related to, and we do see the flagellated stages in animals and choanoflagellates that represent a clade with fungi as defined by DNA sequence analysis. So even though they lack a flagellum, they are opisthocons. They have just shed the flagellum as part of the derivation of some of the clades within kingdom fungi. So the loss of the flagellum, if we want to use our correct technical term, is a derived state or a derived trait of 3 of the major clades that we identify in kingdom fungi. So it's a derived trait, but it is not a derived trait of all fungi. Anyway, let's move on. So the current phylogeny of fungi is one of those things that has been subject to a lot of change over the course of about the last 20 years or so. There has been a lot of rethinking about relationships amongst fungi, what the definition of a fungus should actually be, where certain types of fungi should be placed within the phylogenetic tree. This has all been the subject of a lot of discussion over the last 20 years or so. Where we're at right now in terms of the phylogenetic tree as presented in the textbook is a major reorganization. As I mentioned at the beginning of this presentation, it's a major reorganization of the kingdom, but it seems to be one that most mycologists are clustering around. There seems to be a consensus that this is where the phylogenetic tree should go. So what I'm going to do as we talk about this, about the phylogeny, and as we talk about the different clades, is reference where these clades have been previously, because that is a reflection of some of what you have seen in the fungi lab, and we are actually going to introduce into the next edition of the lab manual the alternative phylogeny as presented in the textbook right now, because not all textbooks are utilizing the same phylogeny. Our textbook happens to use this new phylogeny, whereas other textbooks that you may encounter or have encountered at Valencia or elsewhere are utilizing the older version of the phylogeny. So we want to make sure that we present both phylogenies to you. So you've seen the older phylogeny in the lab manual as it exists right now. What I'll do as I go through the new phylogeny is talk about where the clades within the new phylogeny were previously positioned or not positioned, as the case may be, in the old phylogeny, so that you have a way of making the connections. So this is what we think is the best current phylogeny of the fungal kingdom. So the cryptomycetes and the microsporidia are recent additions. And in the case of the microsporidians, there has been an ongoing discussion as to whether they are true fungi or not. The DNA evidence suggests that they are at least very closely related to fungi if they are not true fungi. You can see that you've got the dotted line. So the dotted line means that this is tentative. If it turns into a solid line, then that means that the people who are doing the taxonomy of the clade and thinking about where everything should be placed are now confident that that's where it should be. And so the dotted line, that tentative connection, now becomes a solid line, a firm connection. So the chytrids have been placed basically to the rest of the definitive fungi, and they are the one clade that is definitively fungi that does have a flagellated stage. They have at times been interspersed amongst an old clade that was made up of the zoopagomycetes and the mucoromycetes, which was previously called, let me erase that. So if you are thinking back to what you saw in the lab manual, you would have encountered the zygomycota, the zoopagomycetes and the mucoromycetes, or mucoromycota and zoopagomycota, are two new phyla that have been created by splitting the zygomycota. So the consensus right now is that zygomycota is no longer valid, that these two fungi really do, or two clades of fungi really do exist on the basis of DNA analysis. And so they have been assigned. And so we now have these two new clades. The ascomycetes and the basidiomycetes, those are still valid. Those you'll find in the old taxonomy as presented in the lab manual. They are still valid today. We still recognize the ascomycetes and the basidiomycetes. So there have been some changes, and we need to recognize those changes and take those into account as we talk about the phylogeny. So the cryptomycetes and the microsporidians are considered to be a sister group and very much basal to fungi. And again, it's a tentative association. So I don't know very much about these because they have been recently identified. I know a bit more about the microsporidians than I do about the cryptomycetes. The cryptomycetes are primarily parasitic on the basis of what I've seen in the literature. They do have a wide distribution. The actual number of cryptomycetes in the environment is uncertain. 30 species have been described, so we say that 30 species are known. But one of the ways that people explore environments for the presence of new organisms is not to look for the organisms themselves, but look for their DNA signatures. And it appears that in some environments, you can identify on the basis of DNA signatures a wide diversity of cryptomycetes. So it's very possible that there's a larger diversity of cryptomycetes out there than these numbers present. It's just that these are being identified on the basis of their DNA signatures, not on the basis of identification of the organisms themselves. So this is a image taken from the textbook here. You can see the spores of the cryptomycete acting as a, I guess, parasite at this point inside a host fungus. You can see the hyphae of the host fungus. So they're actually parasitizing fallow fungi. They do have a flagellated stage. They do produce chitin as part of their cell wall as typical fungi. That's pretty much all we really need to reference as far as the cryptomycota are concerned. Microsporidia are a little bit more important because these are parasites of animals, and they are very much parasites of humans. And like the vast majority of parasites, they are also opportunistic. And so we do see the evidence of microsporidial infections in individuals who have compromised immune systems. And so that's one of the things that we need to remember about microsporidia. What is evidence of their role, of their parasitic nature, is the fact that, like parasites that we've seen before amongst the protists, they have either very reduced mitochondria or non-functional mitochondria. And again, the reason why they don't need functional mitochondria is because as a parasite, they're tapping into the energy resources of the host. So this is, again, a reflection of the microsporidia as parasites. We see that they adapt to this parasitic life cycle by discarding the need for mitochondria because they can tap into their host's energy resources. So here you see an animal cell infected by microsporidians. So here is a more mature individual. Here is the spore. And so you can see that you have a cluster of mature forms plus the spore cells infecting the same host cell. And if you want to put them on a scale, this is a mitochondrion. There's another mitochondrion over here. So let me just circle that and label it because the textbook did not. There's a mitochondria. So that gives you an idea of the scale. These are very, very small organisms. And as a consequence, they also have very small genomes. And some of them have genomes with as few as 2,000 genes in them. And as a consequence, these are also subject to a lot of research, as are the mycoplasmas amongst the bacteria, because of the fact that they have these very, very small genomes with a very limited number of genes, trying to figure out what is the basic set of genes necessary for a functional cell. And so microsporidians are part of that research enterprise as well. So the one thing that sets microsporidia aside from the other basal groups of fungi is that they don't have a flagellated stage. Their reproductive cells, their spores, instead use a harpoon-like feature to allow them to penetrate into the host cell that they're infecting. But again, that's a feature of them as parasites. They focus more on being able to infect host cells rather than being able to move freely through the environment. So it's a fair bet that this harpoon-like organelle is either a derivative or a modification of what was once likely a flagellum that has been modified to allow for better penetration into the cells that they're infecting. The next group are the chytrids. These are the most basal true fungi that are definitively categorized as fungi, and no tentative placement here. These are definitively fungi. These are the fungi that were once classified as protists on the basis of the fact that they do have a flagellated stage, and fungi are not supposed to have a flagellated stage traditionally. The position of chytrids has gone backwards and forwards a little bit over about the last 10 years. So when we had the clade zygomycota, it was somewhat uncertain, would be the best way of putting this, as to whether the chytrids and the zygomycota should be classified as separate clades, and therefore the loss of flagella was a single event that occurred between the chytrids and the emergence of zygomycota from a common relative with or a common ancestor with chytrids, or whether the chytrids should be dispersed amongst the zygomycota, because there was some DNA evidence that suggested that might be a better phylogeny as far as the chytrids with respect to the zygomycota. But it appears that the current consensus now is that chytrids do represent a single flagellated clade, and that the loss of flagella was a single event that occurred after the emergence of chytrids as a clade from the common ancestor that chytrids share with the rest of the fungal kingdom. So chytrids are now considered to be a definitive clade on their own, and the only members of the definitive fungi that have a flagellated stage. The vast majority of them are unicellular, but some of them also form colonies. And where they are currently very significant, and we'll talk a little bit more about these towards the end of the presentation, is that they are very significant parasites in aquatic systems, and particularly parasites of amphibians, salamanders and frogs. And one of the things that we have recognized around the world recently is that amphibians are in danger of becoming extinct in some locations. And when we look for the reasons why, it's chytrids. It's a condition called mycosis, but more of that to come. So here you can see a picture of one of the unicellular chytrids, and you can see the hyphal structure growing out, very classical fungi. But then you have a cell wall around the outside of the cell that's made out of chitin. Again, very classic fungi. Then we have the two clades that are now considered to be two separate phyla that are derived from the old phylum Zygomycota. These are the Zoopagomycota or Zoopagomycetes, and the Mucoromycota or Mucoromycetes. So the Zoopagomycota represent a clade of around 900 species. They can be commensal, meaning they infect animals, but they cause no damage to the animal. They cause no side effect, no ill effect to the animals, but they get a benefit from being associated with the animals, or they can be true parasites. They do produce filamentous fungi, they do reproduce asexually, and they don't have a flagellated spore. So they're meeting all the guidelines of atypical fungus. Some of the organisms that they are known to parasitize amongst the animal kingdom are insects. And one of the interesting things that they do is secrete hormonal-like substances that alter the insect behavior. And there are several pictures available on the internet of ants that have these little fungal fibers. They're actually sporangia or zygosporangia emerging out of the body of ants and so forth, that clearly indicate that the animal is totally riven through by fungal hyphae, and these reproductive structures, the zygosporangia, have emerged. And what is interesting is that these hormonal substances that they're producing suppress normal insect behavior and alter insect behavior. And typically that alteration of their behavior is in favor of the fungi's reproductive approach. And it typically means that the insect will move to a location which under normal circumstances it would never go to because it would put the insect in danger of being eaten. But it's a region that favors the ability of the fungus to reproduce, either because it is going to be consumed by something that will consume the insect, or it means that the fungus can release its spores into the air in such a way that they're more likely to be carried over a longer distance. It gets them up off the ground. Either way, it does not favor the insect, it favors the fungus. And this is not an unusual thing amongst fungi. There are other types of fungi that do this from other clades as well. It just so happens that these types of fungi, when they produce these reproductive structures, produce masses of them that are very, very obvious emerging out of the body of the insect. So whether the fungus itself kills the insect or whether it puts it in a position where the insect is consumed as part of increasing the reproductive likelihood for the fungi, it really doesn't matter. The insects are going to be killed as part of the process. So you get this structure called the zygosporangium that develops typically out of fusion of two hyphae being produced by two mating strains of fungi, the plus and minus mating strains. We'll see this actually in the life cycle of a typical mucoromycete, Rhizopus. So Rhizopus actually belongs to the mucoromycetes these days. And we'll see the origin of the zygosporangium. If the sporangium is produced as a consequence of asexual reproduction, it is called a zoosporangium or a sporangiophore. Both names have been used. And there the spores are produced as a product of mitosis because this structure is already haploid. So you're just going to produce haploid spores from haploid nuclei already present in the reproductive structure, the zoosporangium or the sporangiophore, depending on the type of fungi that it's associated with. So the mucoromycetes are a pretty diverse group of organisms. They represent the remainder of the old phylum Zygomycota, which, by the way, originates from, or the term Zygomycota is a reference to the presence of the zygosporangia, which was considered to be the typical sexual reproductive structure. And we just see those in the Zoopagomycota. And so we see some of those here also in the Mucoromycota. So let's take a look at the life cycle of Rhizopus as a typical mucoromycete. So the asexual reproductive strategy is very straightforward. You get a sporangium produced that releases spores. Those spores then germinate to produce the mycelium. Specialized hyphae are produced at the tip of which develops a sporangium that has a reproductive, has a environmentally resistive coat around it. Within that structure, haploid spores are coated in a environmentally resistant coat, forming the spore cell that is going to be released into the environment when the sporangium bursts open. Those spores then germinate when they land in a suitable environment, give rise to a new mycelium. So we just have a cycle of going around and around, producing the sporangia, releasing spores, producing sporangia, releasing spores. This is the mold-like behavior of these particular fungi, and molds are often seen amongst this group. And so here is Rhizopus growing as a bread mold. Black bread mold is a member of the genus Rhizopus, and you can see the sporangia here on the surface, on the ends of the specialized hyphae that are being produced from the mycelium. If they're going to reproduce sexually, this is where we're going to get the zygosporangium. So we need the two different mating types, the plus and minus. The mating hyphae then are reproduced in response to the release of pheromones from each of the two mating types, and they grow towards each other, and each of them contain copies of the haploid nuclei of each of the two of the mating strain that is producing this reproductive hyphae. When they encounter each other, they produce specialized gametangial structure that will then ultimately fuse to form the zygosporangium. That zygosporangium then gets coated in a environmentally resistant coat. It remains as a stable structure for a indefinite amount of time. Eventually, the two haploid nuclei fuse via karyogamy, giving diploid nuclei. Those immediately undergo meiosis. The resulting haploid nuclei then get encapsulated into a cell structure that gets coated with a environmentally resistant coat. Those are then released as spores to germinate into the new mycelia, and you get equal numbers of plus and minus mating strain spores being released in the environment to maintain the continuity for sexual reproductive purposes, should that be required. So these are identified on the basis of this reproductive sexual strategy involving the zygosporangium. And remember, the old phylum that included both types, the Mucoromycota and the Zoopagomycota, were once part of a single phylum, the Zygomycota, because representatives of both clades produce the zygosporangium, the sexual reproductive structure. So there's the connection to sexual reproduction and the old clade name, Zygomycota. Now, some members of the Mucoromycota, and I've got a picture of Polobolus coming up, are actually able to distribute their spores, not randomly, but deliberately in one particular direction. So Polobolus grows on cattle dung, right? Cow pies or cow pats, however you want to call it. And so in order for their spores to wind up in the cow dung, they actually need to be consumed by the cattle, by cows. And so Polobolus adopts a strategy of having a sun-sensing organ on the tip of the sporangium, and it tracks the sun. And the reason why it tracks the sun is because cows eat grass. Grass grows in the sun. So if you want to increase the likelihood of your spores being eaten by cows or other grass-eating herbivores, so it could be deer, antelope, things like that, anything that eats grass, you want to get your spores into a sunny area because that's likely where the grass is going to be. So the sporangia track the sun and shoot the spores in the direction of the sun, because that's where the grass is going to be. That's where the ungulates, the deer, the antelopes, the cows, horses, whatever, are going to eat the grass, ingest the spores. The spores are coated to resist stomach acid and pass through the digestive tract. They get excreted with the manure, and they now then germinate on the manure and develop into the new mycelia growing in the excuse me, in the manure and breaking down the undigested and partially digested plant material that is part of the cow pat or the dung of whatever animal excreted it. But this is how Polobolus guarantees that its spores are going to wind up in the right place, in amongst the grass, in a sunny spot where they're most likely to be eaten. So you can see different types of complex behavior amongst the fungi. We're going to see a few more of those along the way too. So Glomeromycetes are a group of around, I think the number was originally pegged at around 30 to 50 species of what used to be zygomycetes. So about 10, 15 years ago, what came to be known as glomeromycetes were actually an integral part of Zygomycota. Then they were separated out and given their own phylum, Glomeromycota, on the basis of the fact that these are the fungi that form the arbuscular mycorrhizae with plant roots. But these have now, with the splitting of the Zygomycota into the two clades that we've just been talking about, the Glomeromycota have now been reclassified as Muromycota. So they're back amongst the fungi from which they were originally split when Zygomycota was the clade name for all of these. So just to remind you again, the arbuscular structure is that very highly branched hostorium that is formed within the confines of the plant cell wall, but does not penetrate through the plant cell membrane. And what this does is provide a large amount of surface area between the fungal hyphal mass and the plant cell membrane for the transfer of materials backwards and forwards between the fungus and its plant symbiont. So this is the hostorium. Let me just make sure the pen is active. So this is the hostorium. And this structure here, this is the cell wall. So these fungi produce a very active enzyme called cellulase. They have a particularly good form of cellulase that can digest its way through cellulose, the plant cell wall, and allow the formation of this hostorium within the body of the plant cell, or within the confines of the plant cell wall, but not within the plant cell proper. And that cellulase enzyme is now subject to some investigation as a potential way to increase our ability to make biofuels. I'll talk a little bit more about that at the end of the presentation when we talk about some roles for fungi. The next clade, the Ascomycetes, is one of the clades that has survived the rewriting of the phylogeny of the fungal kingdom. This is still considered to be a valid clade and a monophyletic clade. These are sometimes referred to as sac fungi because of the presence of a sac-like structure called an ascus, asci being the plural. That is the fruiting body, or is the source of the spores within the fruiting body, called an ascocarp. That is common to all Ascomycetes. So the fungi that you might be familiar with that come out of the clade Ascomycota are these morels, which we do consider to be edible, and they are somewhat expensive. They usually sell in markets for $10, $20, $30 a pound. They're considered to be somewhat of a delicacy. But the ones to the right are the ones that are really expensive. So these are the black truffles. And these usually sell for hundreds of dollars per pound, but these are not the most expensive of mushrooms or the expensive fungi. The really expensive fungi are the white truffles. Now, I happen to have a black truffle in our collection of materials for the fungi lab. You should have seen a picture of it. We've tried to include pictures of all the relevant fungi in the fungal lab, and I believe we put in the black truffle, but I have never had a piece of white truffle, nor do I ever expect to have one. I was very fortunate in that a former student a number of years ago had a whose family owned a restaurant, bought in a small black truffle after we'd discussed fungi, and he said, "I'd like to donate this to the department and to the class so that students can see a black truffle." And that little piece of black truffle was worth quite a bit at the time. It's worth even more, but it's under preservative, so no one can ever use it. What about white truffles? Well, white truffles can sell for thousands of dollars a pound. In fact, the most expensive white truffle to be sold was a piece of white truffle that weighed about 1.5 kilograms, so around 3 pounds. And it sold for around $325,000. Was going to a restaurant, and the owner of the restaurant, who was an Arab sheikh, decided that he wanted to have this white truffle to utilize in his restaurant, and he was willing to pay the money for that. $325,000 for about 3 pounds of white truffle. And you might ask, why are they so expensive? And the reason is because they are so difficult to find. These fruiting bodies are strictly produced underground by fungi that are symbionts of the roots of oak trees in Europe predominantly. Now, we do have truffles that are native to the Pacific Northwest, but they are nowhere near as expensive as these, nor are they considered as valuable cuisine-wise as the white and black truffles. Those are the really expensive ones, and those are the ones that are really valued by chefs of high-end restaurants. The reason why is because they have a very intense flavor, and you only need a small amount in order to flavor a dish. And because they are so difficult to get, because they're underground, they only grow in very specific regions, and up until very recently could not even be grown commercially, because no one could figure out how to synthetically or artificially seed the roots of oak trees. That's now been accomplished, and a couple of countries now have black truffle industries, but no one has yet successfully managed to commercially develop artificial white truffle production. It still relies on natural production of white truffles. And so because these are incredibly rare, incredibly difficult to find, they are very valuable. And if you are lucky enough to have a dish that contains a little bit of white truffle or a little bit of black truffle, savor it, because these are considered to be very high-end delicacies and extremely expensive. So if you ever get an opportunity, if someone offers you an opportunity, I would grab it. I've never been given an opportunity. One of these days, maybe I'll pay for it myself, just so I can say that I've experienced it, but up till now, no such luck. Anyway, ascomycetes also represent a lot of the molds that are considered to be agricultural pests. They're also important decomposers. They're also important symbionts. So they serve a number of roles. When they reproduce asexually, they produce the conidia on the tips of the conidiophore. So we looked at those on the surface of the orange in a previous diagram, and that's where we were talking about the yeasts as well. So yeasts are tentatively placed amongst So yeasts are tentatively placed here. because they produce that bud that looks very much like a conidium, which is characteristic of the asexual reproductive stage of the ascomycetes. So this is where yeasts are being tentatively placed, even though we don't see their sexual reproductive strategy very often. It's very infrequent that yeast will reproduce sexually. They predominantly reproduce asexually. Another member of the ascomycetes that you may be familiar with is Neurospora crassa. This has a history in terms of our understanding of information flow in cells, the idea of DNA to RNA to protein, because it was using Neurospora crassa as a model organism that the connection between mutations in DNA and loss of enzyme activity was established by creating artificial mutants that required supplements to the media that they were growing on, and recognizing that the loss of growth on minimal media and the requirement for supplements was a product of loss of a single biochemical step, which meant that it was the loss of a single enzyme, hence the connection between a mutation in a gene and the loss of the enzyme activity. It became known as the one gene, one enzyme hypothesis that would ultimately develop into the one gene, one polypeptide concept that you hopefully learnt in Biology 1. So this is the life cycle of a typical ascomycete. So let's begin with the mycelium and take a look at the asexual strategy. So the conidiophore is the product of a specialized hyphen that emerges from the hyphae that make up the mycelial body. At the tip of the conidiophore, the haploid nuclei are packaged into individual cells. These become the conidia that are then released as spores into the environment. When they land on a suitable food source with moisture, they germinate to give rise to the new hyphen that will develop into the mycelium. If they are going to reproduce sexually, then the conidia of the minus mating type lands on the end of a hyphen of a plus mating type. This then gives rise to a structure on the end of the hyphen called an ascus. This is the sac arrangement. This then develop, or sorry, I should back up a minute. The ascus will develop as a consequence of this fusion, but what initially develops is the dikaryotic hyphen. And I ought to be a little bit more definitive here about the dikaryotic hyphen, because I mentioned that back when I was talking about the general life cycle. So I really ought to address this a little bit more succinctly. So the fusion of the conidium, the minus type conidium, with the plus type hyphen gives this dikaryotic stage, because the hyphae are split up into cellular chambers or cellular compartments by the septae. And so you have one copy of each of these haploid nuclei present in this hyphen. If it continues to grow, which it can do, not in all of the ascomycetes, but in many ascomycetes, this hyphen will continue to grow in its dikaryotic form. But at some point at the end of the hyphae, branches will occur to give rise to the ascus. Still dikaryotic, still two separate haploid nuclei. But within the ascus is when the haploid nuclei will fuse to give the diploid nucleus, which gives you the zygote. Immediately, this haploid nucleus then, diploid nucleus. Gee, I'm having trouble keeping those straight today. The diploid nucleus then divides by meiosis to give 4 haploid nuclei that arrange themselves in a linear fashion inside the ascus. each of these nuclei then undergo a mitotic division, so that you now have not 4 haploid nuclei, but 8 haploid nuclei arranged in a linear fashion inside the ascus. Each of these haploid nuclei then gets compartmentalized into an individual cell, which becomes a spore. Now, here we have the possibility for sexual recombination, because during meiosis, you have the reshuffling of the genetic material. You should remember that as part of your Biology 1 course. You learned about meiosis and the fact that you have chromosomes being recombined as part of meiosis. So you get origin of genetic diversity, and then you have that followed by mitosis. And so now you have an opportunity to look at the way in which sexual diversity or genetic diversity can be a product of both meiosis and mitosis. And this has meant that not only were organisms like Neurospora crassus important for establishing the connection between genes and gene products, proteins, but they're also now turning out to be very important in understanding the role of meiosis and mitosis in creating genetic diversity through distribution of chromosomes. We know that there is genetic diversity as a product of meiosis, but it turns out that mitosis may not be as non-random as we might have previously considered, or may not be as random. There may be some non-random distribution of chromosomes during mitosis as well. And organisms like Neurospora crassus provide opportunities to investigate that. So these fungi have become important because we know the origins of those 8 haploid nuclei. We know where they come from in terms of meiosis, this linear arrangement inside the ascus. And when they reproduce, the 2 daughter haploid nuclei as product of mitosis are sitting next to each other. So now we know the direct origins of all that 8 nuclei, and we know which are sister nuclei. And so we can take a look and see what sort of patterns of genetic diversity we see in those 8 haploid nuclei as a product of meiosis and mitosis consecutively. Stumble on that one. Anyway, the asci are contained within the ascocarp, the fruiting body of the ascomycetes. Once one of the ascocarps ruptures and releases the ascospores from the asci, all of them rupture. So individual asci will rupture, but that triggers all the asci to rupture, which means that all the asci within the ascocarp rupture. And in the case of some of these ascocarps, apparently it's audible. I'm informed by some people who have apparently heard this, that in some cases it's loud enough that it sounds like falling rain. It's quite distinctive sound, apparently, because all of these asci are rupturing all at once and releasing the spores, and it creates an audible sound. The last major clade are the basidiomycetes. These are the ones that most of us are familiar with because these are the ones that produce mushrooms. These are also the fungi that have no known asexual reproductive strategy. They reproduce strictly sexually. And for this reason, their life cycle is kind of interesting to look at, because they form not a transitory diploid stage in the traditional sense, but they form a long-lived heterokaryotic phylum that contains multiple copies of the 2 haploid nuclei. And that can exist underground for years, centuries, without producing a fruiting body. When it produces a fruiting body, we refer to that as a mushroom. So when you eat a mushroom, what you're actually eating is the fruiting body, the reproductive structure of a basidiomycete. The other type of fungi that traditionally are included amongst the basidiomycete that you might be familiar with are the shelf fungi. So if you ever see something like this growing on the side of a tree, that tree is doomed, because shelf fungi have a very interesting nutritional life cycle. When they first infect the tree, they infect a living tree. So they're strictly parasitic, and they will over time kill off the tree. But they're not done with the tree at that point. They're not going to go on and look for another living tree to parasitize. No, the shelf fungi now switches over to what is referred to as saprobism. They become saprobes. And a saprobe breaks down dead tissue. So they have two different nutritional modes within the same life cycle. They begin as parasites and eventually wind up killing the tree that they're growing on, and they grow traditionally on trees. Once they kill the tree, then they switch over to saprobism and start breaking down the dead material. So they make full and complete use of the tree that they have infected. Puffballs, well, if you've ever seen a puffball, you've probably kicked it to release the quote unquote dust. Well, that dust happens to be their spores, and you've done exactly what the puffball wants you to do. Release its reproductive spores into the environment so that they can reproduce again. The last one that we've got here in the diagram, the maiden veil fungus, is really interesting because this produces the same chemical that gives rotting flesh its odor. So these literally smell like dead stuff, dead animal. And sometimes when you smell the smell of dead animal in the bush, in the scrubs, you're not actually smelling a dead animal. What you're smelling are these fungi. And you might ask yourself, why would a fungus need to produce the same chemical as a dead animal? Well, if you think about what gets attracted to a dead animal, the first thing that you probably think of is flies. Because flies are attracted to dead animals. They lay their eggs in the rotting flesh. And when the maggots hatch out, they consume the rotting flesh. What the fungi are doing are attracting the flies. Why? Because they want the flies to walk all over the fungus and pick up the fungal spores and transport those to a new location so that the fungi can, or the spores can germinate to give rise to a new mycelium. So all the fungi are doing here, what these maiden veil fungi are doing, is tricking the flies into picking up the spores and acting as a spore distributing vector to get those spores to where the fungi want them, typically associated with rotting flesh. And so in order to get the flies to come to them, they need to smell like something that the flies are looking for, dead animals. And so they produce exactly the same chemical. An interesting interaction between animals and fungi. We don't necessarily associate animals as a spore distributing vector for fungi. We do think of them as a distribution vector for plants, but here's a fungus doing exactly the same thing. So when we start taking a look at the life cycle of a typical basidiomycete, we're going to see a strictly sexual-based reproductive strategy. So here's the life cycle. So we'll start with the new mycelium germinating from spores that have been released. And again, we're talking about opposite mating strains, plus and minus mating strains. They produce specialized reproductive hyphae that grow towards each other, again in response to released pheromones. the hyphum fuses. And that hyphum then produces, again, a dikaryotic mycelium that is segmented into individual cells, each containing a copy of the 2 haploid nuclei. Unlike the ascomycetes, where this is somewhat transitory, in basidiomycetes, this can be long lived. In fact, the vast majority of those honey oyster mushrooms in Oregon that underlie that aspen grove in the forest, that is one of these long-lived dikaryotic mycelia that give rise to the mushrooms, because it's this dikaryotic mycelium that actually produces the mushrooms. And it can do so literally overnight. I clearly remember on many occasions when I used to live in a house, going out into the backyard, right time of the year, looking around in the grass, no mushrooms. Come out the next morning and there are mushrooms in the grass. They have literally emerged overnight. We don't know what the environmental signal is that triggers this, but some environmental trigger does signal the development of the mushrooms as the fruiting bodies. They emerge as a very dense clump of these dikaryotic mycelia that are forming the stem and the cap of the mushroom. This is strictly referred to as a basidiocarp, and it is dikaryotic, but it is functionally haploid, even though it's carrying 2 haploid nuclei. It's functionally haploid because the two haploid nuclei are still individual nuclei. Within the cap, you have this highly folded structure developing called the gill. On the surface of the gill, you have the formation of these club-like structures called the basidia. And so these are sometimes referred to as club fungi because of the presence of the basidia. Within each basidium are the 2 haploid nuclei, 1 copy of each of the ones that were derived back here when plasmogamy occurred and these 2 reproductive hyphae merged to give rise to the dikaryotic mycelium. These basidia are products of that dikaryotic mycelium, so they carry the 2 haploid nuclei. It is within the basidia that you get karyogamy within the sealed mushroom. So there is a membrane that covers the gills. This is all happening in the sealed mushroom. Karyogamy occurs, you get the diploid nuclei. That immediately undergoes meiosis to form the 4 haploid nuclei that then migrate to the tip of the basidium and get encapsulated into individual cells that are attached to the end of the basidium. These become the basidiospores. When all of the basidiospores are mature and ready for release, the membrane dries out and releases or retracts and uncovers the gill structures on the underside of the mushroom cap. This results in the release of the spores, and they get carried by the wind for dispersal into the environment. When they settle, they then germinate if the environment is suitable and start forming a new mycelium, eventually to hopefully find a member of the opposite mating strain and form a dikaryotic mycelium to continue the process. Now, if you want to see the release of these spores from the mushroom, if you buy mushrooms from the supermarket, say some of the large portobello mushrooms, with the membrane still intact, so you can't see the gill structure on the underside of the cap. If you then cut the stem off so that you can lie the cap on a sheet of paper with the gill side down, you just sit and wait. Eventually, this process will play out so long as the mushroom is kept in a suitable environment. You want to keep it relatively moist, and it's only going to take a day or 2, so it's not going to dry out particularly quickly. Eventually, the membrane will dry out and retract, exposing the gills. The spores will drop and they will land on the paper. If you then carefully remove the mushroom cap, you can see outlined in the spores deposited on the paper the pattern of the gills that were present in the cap of the mushroom. You have to be a little bit careful here, because if you blow too hard or set up an air current, you'll disrupt all the spores, because they are designed to be carried in the air, so they are very light and it's very easy to disrupt them. But you can see that pattern formed by the spores dropping onto the sheet of paper if you're really careful with it. So this is our next concept check, and I'm just trying to think. Yes, I should take a break here because we've been going for a little bit of time at this point, because we've been laying out the origin of mushrooms. We've also talked about the phylogeny, and we've talked about the various clades. So we should probably take a break at this point. So here are the two concept check questions, and I'll see you on the other side of the break for the answers to these two questions. So what features of chytrids suggest that they are members of the basal fungal lineages? And there's one feature that's really obvious for that. How does form follow function in the mucoromycetes, the ascomycetes, and basidiomycetes? I'll see you on the other side of the break with the answers.